Good morning. So we'll start our class today with Phylum Porifera, or the first phylum in Kingdom Animalia. Okay. Now, actually, in case of Animalia, we can say that this is a phylum which is basically made up of only cells. Okay. And that is why they are called a loose aggregation of cells. So, phylum Porifera. Now, what phylum Porifera means? Now, in Greek, okay, poros means holes. In Greek, poros means holes plus pherein means bearing. So they are called pore bearing animals. So poros means holes and pherein means bearing. Okay. So they are commonly called pore bearing animals. Okay, they are called pore bearing animals. Okay, so this is our first phylum, and since there is no notochord, so they are also referred to as non-chorded phylum. Okay. Now, so let us have some discussion about their history. Okay, history we will discuss. So earlier, these animals, okay, so a lot of them thought that these are plants. Fine. A lot of them thought they are plants. They look like plants. Okay. But later on, the idea changed. So, now, the first person, as you know, in the old Greek people, so Aristotle referred sponges as animals. You see, there is something about Aristotle that he was so correct in a lot of his statements. Okay, so Aristotle believed that sponges are animals. After him, people thought that they are plants. Okay, referred sponges as plants. Now, why they refer sponges as plants? You can have some features. So, they are fixed, that is sessile or sedentary. Fine. Number 2, they show vegetative propagation. So if you break a sponges into many pieces and throw in different directions, so they will produce many sponges. Okay, so they show vegetative propagation. Now, a lot of them are green in color. Okay, but green color is not due to chlorophyll actually. But the green color is not due to chlorophyll. It is due to the association. Okay, it is due to the association of zoochlorella. Okay, it is due to the association of zoochlorella, which are actually unicellular Okay, unicellular green algae. You understand? So that is why. So due to so many features, they are actually referred to as what? They are actually referred to as plants. Now, Ellis classified them as animals. So he pointed that sponges are animals. Okay. Linnaeus, Lamarck, and Cuvier placed them in Coelenterata. Okay, they placed them in Coelenterata. Okay, in zoophyta. Okay, so zoophyta means plant-like animals. Now, in 1836, Robert Grant proved sponges to be animals. Okay, he proved that sponges are animals, and he created a new phylum that is called Porifera. So he classified them in a separate group known as Porifera. Okay, Huxley and Sollas proposed complete separation of sponges from other metazoans. Why? Because what are metazoans? Animals with definite tissues. Okay, so Huxley and Sollas proposed that sponges are actually not metazoans. So they propose that they should be removed from the metazoa category. Sollas placed them in a branch which is called parazoa. So what are parazoa? So these are animals which show cell aggregate body plan. Okay, so these are animals which show cell aggregate body plan. Okay, so that is the history of this category of animals. So many very important discussions we have discussed. Okay, now उसके बाद हम लोग discuss करेंगे is let us introduce them. So what is their introduction and definition? So we will place them. Okay. Now introduction. So how do we introduce Porifera? They are pore bearers. They evolved from colonial protozoans. Okay, which are the colonial protozoans? They are actually called choanoflagellata. Okay, so they are called choanoflagellata. Or इसमें example you can know Proterospongia. Okay, उसका example है Proterospongia. We call them connecting link. Okay, connecting link between. Okay, so they are called connecting link between unicellular and multicellular animals. Okay, so they evolved from choanoflagellates. Example is Proterospongia, and now we refer them as connecting link between unicellular and multicellular animals. Okay, and thus they are more complex than sponges. Okay, than protozoans. Actually, these colonial protozoans, they are more complex than protozoans. Okay, now, so that is why they are called pore-bearing animals. Now, what is their definition? The definition we will say. Okay, so the name was coined by Robert Grant. Okay, as you know. Now, it includes about 10,000 species, both living and fossils, belonging to 1400 genera. So this data may vary if you see books to books. The reason is who is referring to what? So they are commonly called sponges. Why they are commonly called sponges? They resemble the chemical sponge in your house, okay, which is used to make cushions, etc. Okay, so since they have resemblance with it, they are commonly called sponges. Now they are grouped under the group Parazoa, okay, and so their study is called parazoology. Okay, so their study is called parazoology. Now, in case of their definition, so we can define them. Fine. So how we will define them? तो आप देखेंगे animals with cell aggregate body plan, cellular, okay, cellular level of organization, okay, cellular level of organization, hence no organs and no organ systems. Okay, so since there is cellular level of organization, tissues भी बना नहीं है, तो organs भी नहीं होगा and organ systems भी नहीं होगा. Okay, so animals with cell aggregate body plan, cellular level, hence no organs and no organ systems. उसके बाद और क्या होगा? So sessile, fixed to substratum. Okay, they are sessile, fixed to substratum, means वो जमीन में fixed है. Okay, next. So what are the other features? Okay, characterized by the presence. Okay, characterized by the presence of choanocytes, spicules, and a definite canal system. Okay, so this is a very, very important point about your porifera. Fine, so they are characterized by the presence of a definite canal system. Okay, other features you can say that gemmules for asexual reproduction. This is another special feature. Gemmules, they make gemmules for asexual reproduction, and both solid and hollow larva. Okay, so these are the definition. This is how we define these animals. Okay, so now we will go into the general detailing. So whenever we are doing general detailing, so we will start our points from your general features. Okay, and we will try to fill up a form. Okay, so we will try to fill up a form. So that is why in case of your general points, always we'll try to fill up a form so that writing taxonomy becomes easier. Okay, so writing taxonomy becomes easier. So the first point that we are discussing is general characteristics of porifera. Okay, so usme jo habit and habitat means where do they root, where do they live? They are commonly known as sponges. They are aquatic, sedentary, mostly marine. Okay, they are mostly marine, but a single, okay, but some found in freshwater also, So they belong to a single family. Okay, they belong to a single family known as spongylida. Okay, they belong to a single family known as Spongylidae. Okay, now example include Spongilla and Ephidatia. Okay, Spongilla and Ephidatia. So these are freshwater sponges. Okay, you can write fresh water sponges. Understood? Now these individuals are sessile, or you can say immotile. Fine? And they are attached to the substratum. They are all attached to the substratum. So earlier, okay, so earlier people mistook them as plants. Okay, so this is the reason they were thought to be plants. Now they contain flagella in their collar cells. So these cells are called collar cells or choanocytes. Collar cells bracket रे आप लिख सकते हो characteristic cells of these animals. Okay, characteristic cells of these animals, they are also called as choanocytes. Okay, collar cells or choanocytes. Fine. So which are characteristic cells to these animals. Fine. Now these cells are responsible to bring in water, and with water comes food, oxygen, food, oxygen, gametes. Okay, along with water goes, क्या बाहर चला जाता है? Wastes, CO2 and gametes. So that is why water is very, very essential for these animals. Along with water, they come in and they also go out. Okay? So these cells are responsible for bringing in water, and with water comes food, oxygen, gametes, and along with water goes waste, CO2 and gametes. Okay? Now these cells are present in the spongocoel, okay, and thus set up water current. Okay, so this is the basic concept. Now they are found in warm, shallow waters. Generally, actually, you will always use the term generally, because in biology you will always find exceptions. Generally, they are found in warm, shallow waters, but there are sponges which are also found in deep sea. Okay, they are also found in deep sea. Okay. Now, उसके बाद है body. So now, what is their body? So now the body is diploblastic. They have two germ layers, okay, with an outer ectoderm Okay, made up of pinacocytes. What are pinacocytes? Flat cells. Okay, and so the outer layer is known as pinacoderm. Okay, so outer ectoderm, you can write like this, an outer ectoderm made of pinacoderm. So the outer layer is known as pinacoderm, and inner endoderm made up of choanocytes. So it is also known as choanoderm. Okay, choanoderm. And since it helps in digestion and absorption of food, it is also known as gastroderm. Okay, so it is also known as gastroderm. Okay, so inner layer is made up of choanocytes, so it is called choanoderm, and since it helps in digestion and absorption of food, it is also known as gastroderm. Okay. Now, the space between the two layers is occupied by a non-cellular gelatinous matrix, also known as the mesenchyme or mesohyl. Okay, also known as mesenchyme or mesohyl. Basically, mesenchyme we will not use, we can use mesoglia. Okay, so the space between the two layers is occupied by a non-cellular, yeah, we can write simply by a gelatinous layer, okay, also known as mesoglia or mesohyl. It acts as a glue to join the two layers. Okay, join the two layers. There are some wandering cells. Okay, there are some wandering cells. Okay, there are some which are known as amoebocytes. Okay, which are known as amoebocytes, and they are of different types on the basis of their function. Okay, on the basis of their function. Now, these cells are responsible for various activities like communication among cells and functioning of the sponge. Okay, so that is why here we are basically finding that there are three layers. Okay, two layers, and which is joined by a gum. Okay. Okay, in the middle there is a, okay, fine. So this is the concept. So now we will make a drawing. Okay, we'll try to make a drawing of these cells. Fine. So if you see a general sponge, we'll make a general sponge, and then we'll make a what? We will enlarge the view. Okay, so now suppose this is a sponge body. I am drawing a simple sponge body. Fine. So this is a simple sponge body. So this outer layer, which is blue in color, okay, is also called the pinna coda. Fine. So this outer layer is called the pinna coda. Fine. Then we will have a mesoglea. Okay. So this is the gum-like layer. Fine. So this is the gum-like layer. And inner to this, we have the choanocyte layer. So this is the choanocyte layer. Fine. So now we have 3 layers. And now on the top, there will be a net-like structure, which is called osculum. Fine. So this is the vertical section of a sponge. Bracket mein you can write solitary simple sponge. Fine. So here the outer layer or outer pinnacoderm, the middle one, middle mesoglea, and the inner, that is called choanoderm. Fine. So these are the 3 layers. Plus this cavity is called the spongocele or cavity of the sponge. Fine. So this is the base, okay, which is attached to the substratum. Now this hole is called osculum, and this is basically exit. Fine. And this structure is called oscular fringe, or this is a what? This helps to filter out water. So whatever will go, it will go through this fringe and out. Now these openings जो आप देख रहे हो, these small openings, they are called ostia or mouth. Fine. So, or inlets. So we can say that sponges have many mouths and one anus. Okay. Why? This is basically solitary sponges. So in solitary sponges, there are many mouths and one anus. So now we will draw the water route. So suppose this is outside. So water will go and enter into the body through ostia. Then it will go into the spongocele, where it will be treated by choanocytes. So they will check the water, whatever food, oxygen is there, they will take it, and whatever waste there is there, they will throw it, and ultimately this water will pass out of the body via osculum. So that is why we can say that water route. So this canal system which you are seeing, this is called the simplest form. You will find Ascon type canal system. Ascon type canal system. So the water route is outside Ostia, okay, outside Ostia spongocoel, bracket you say, used by choanocytes, then it will go to osculum and then back to outside. Okay? So this is the concept of the sponge body. So here we are just seeing a simple structure. In the next diagram, we will make the detailed view of only one part. Okay. So now I will show you a magnified view of only this part. Okay. I will show you a magnified view of only this part in the next diagram. Okay. So we will maintain the colors. So now you will see that the outer layer is made up of flat cells. Okay, so these are the flat cells of the outer layer. So they are called pinacocytes, and in between them there is a porocyte. So this cell is called a porocyte. Why? Because there is a pore inside it. So water can cross through. So this is the pore. And ये आपका normal cell है. So this is a normal cell. So we can do some shading. Okay. So this is your porocyte, your cytoplasm. Fine. And this is the what? Choanocyte, pinacocytes. Now, in the inner layer, you will find many collar cells. So first we'll make an outline so that we will not cross the limits. So these are the collar cells. We'll make, these are the cells. Okay, these are the characteristic cells of the sponges. Okay, so these are the collar cells. Fine. Now, so this is actually the collar. ये जो structure हम बना रहे हैं अभी, ये collar है. So that is why they are called collar cells. Fine. Now, these cells, they have flagella. So we'll just give them a color. Just to, this is the shading part. Again. So now they have a finite nucleus. Okay, so we'll make a big nucleus. So this is the nucleus of these cells. And there is a flagella in these cells. So this is the flagella. So these cells are also called flagellated cells. Okay. So these cells are also called flagellated cells. So this layer is called the See, so this layer is called pinacoderm, and this layer is called choanoderm. In the middle, this layer is called mesoglea. Okay, why? There is only gum layer plus we can also call it mesenchyme because there is cells. So now let's color it. We colored it yellow actually. So here we will color the mesoglea. Right? We can color the mesoglea like this. Fine. Now this is the mesoglea. Now within the mesoglea, you will find many types of cells, and these are all amoeboid cells. So depending upon their function, we can give them name. So this yellow matrix is called mesoglea. इसमें आप लिख सकते हैं cells plus mesoglea. So mesoglea. Now suppose this cell, it helps to they are called trophocytes or nurse cells. So what they will do? They feed the entire sponge. Okay. Now, so we will make another cell. Suppose we'll make a colored cell. Okay. Suppose we'll make a colored amoebocyte. So this is chromocyte. Chromocyte gives color to the sponges. You will find sponges in all colors. Uske baad you will find, suppose, another cell. So this is called thezocytes. What are thezocytes? Store food. Okay, excess food जो बच जाएगा वो उसको store करेगा. Tapare will make, suppose this is collencyte, produces a collagenous magnet-like structure. So what these collencytes will do, they will produce what? This network. So this is your collagenous structure, and this net-like structure is produced by collencytes. So इससे क्या होगा? They are tying the outer and inner layer together. So now the outer and inner layer cannot be removed. Here we will produce scleroblasts, or which make spicules. So suppose this is a scleroblast. Scleroblast. Now this scleroblast is producing, suppose, spicule. तो देख रहे हो? तो ये जो कांटे जैसा स्ट्रक्चर है, this is called spicule. So the scleroblast produces the skeleton of sponge. Now एक और हम लोग सेल बना लेंगे. So any number of cells you die, the function is different. So this is called archaeocytes. Very important. So they are totipotent cells of sponges. So what they will do? They help in repair regeneration. Repair and regeneration. Okay. So many types of cells you have seen. In the body. So that is why these body cells they perform different functions. Okay, so that is why what happens, the water which is there, now this water will enter into the body through what? It will enter into the body through your pores, and this pore is called what? This pore is called your porocyte. So these all leveling we have done, that is of the amoebocytes. All of them are amoebocytes. But this blue cell on the outer one, so this is called pinacocyte. And the porocyte is a specialized pinacocyte. Porocyte is a specialized pinacocyte. तो उसका जो opening है, this opening is called ostia. So water will go from here into the sponge body. Understood? Now these red cells, they are the characteristic cells called choanocytes or flagellated cells. They have flagella, so they are also called flagellated cells. Understood? Now, so this structure is called collar. Hence, therefore, also known as collar cells. Okay? So, इसीलिए, so in the amoebocytes, we have discussed your collencyte, archeocyte, chromocyte, thesocyte, trophocyte, scleroblasts, okay, and some other cells also. If we remember, we can make. Suppose this is your gland cell. So what it will do? It secretes slime to protect from water. Okay, to protect from water. So what the slime will do? It will protect the sponge body from water. Second karibo sponge ର ଦେହ ବାହାରେ ଗୋଟେ କଣ ଗୋଟେ layer କରିଦବ. So they will produce a layer of, we'll use a green color, different color. So we'll produce a layer of slime. Okay, so this is a layer of slime. So who has produced this layer of slime? The gland cell. Okay, so this layer is called slime. So it is protecting the sponge from water. Okay, similarly एक और cell हम लोग ये बना सकते हैं. This is called germ cell. So ये क्या करेगा? This produces gametes. Okay, so आ गया मजा? So do you like this drawing? So here we have drawn all the cells of the sponge, and what have I drawn here? See this drawing? This one. So this wall, when we enlarge it, this wall, when we enlarge it, इसका enlarged view in next page. Okay? So when we enlarge this wall, we find this structure. Okay? So this is the concept of the body wall of sponges. Okay? So we can say that the sponge body wall, okay, so we here we can write all the layers. Okay. Now the sponge body wall has the following layers of different types of cells. Okay, the different types of cells. So here we will use the different types of cells. Okay, and उसमें हमारा कौन कौन सा cells आ जाएगा? So many cells we will find in the sponge body wall. Okay, now, okay, so A is outer layer or pinacoderm. Okay, the outer layer or pinacoderm. So you will find what? Pinacocytes and porocytes. Right? Pinacocytes, polygonal flat cells that form the pinacoderm. Now, in simple sponges, they form only ectopinacoderm. Okay, but in complex sponges, they form exo and endopinacoderm. Okay, full stop. Endopinacoderm lines all the canals other than flagellated. Okay, so endopinacoderm lines all the canals other than the flagellated canals, और उसको हम रेडियल कैनाल्स बोलते हैं. ठीक है, तो रेडियल कैनाल को छोड़ दो तो बाकी सारे कैनाल को कौन लाइन करता है? एंडोपिनाकोडर्म. Now porocyte, they are the source of ostia. They are the source of ostia or mouth. Okay, they help in the passage of water from outside to inside. ठीक है, तो ये आपका पोरोसाइट हो गया. Now the second layer is the B. Okay, so ये हमारा inner layer है और this is called choanoderm or flagellated layer or gastroderm और एंडोडर्म. तो पिनाकोडर्म को आप एक्टोडर्म भी बोल सकते हो. क्लियर? नाउ सो दिस मिडिल लेयर, ये जो इनर लेयर है, सो दिस इज मेड अप ऑफ व्हाट? कोआनोसाइट्स. राइट? नाउ digestion, these cells have a flagella which generates a whirlpool and draws water into the sponge body via ostia. तो इसलिए ऑस्टिया से पानी आएगा, पर ला कौन रहा है? It is brought by flagellated cells. These cells are the first to receive food and digest them also. है ना? तो ये खाने को रिसीव भी करते हैं एंड डाइजेस्ट भी करते हैं. तापरे the third layer is called mesenchyme. What is mesenchyme? Mesoglia plus free amoeboid cells. है मेसोग्लिया प्लस फ्री एमीबॉइड सेल्स. ठीक है, ठीक है. सो इसलिए तो इसमें आपके आ जाएंगे. सो हियर यू विल फाइंड द एमीबोसाइट्स आर डिफरेंट टाइप्स जो आप देख रहे हो. सो एमीबोसाइट्स है कॉलेनसाइट्स, आर्कियोसाइट्स, क्रोमोसाइट, फिजोसाइट, ट्रोफोसाइट, फैगोसाइट्स. सो ऑल ऑफ देम आर देयर. Fine. So this is the concept. हाँ है. तो now scleroblast जो है skeleton secreting cells, there are 3 types. तो calcoblast ये क्या करता है? Secrete calcareous spicules. Okay. Silicoblast produces siliceous spicules, and spongioblast secretes spongin fibers. ठीक है. So these are the different cells of the sponge body. Fine. So today we will end the class because already 10:40. Okay. So if there are any doubts, you can send in the group. Right. And we will continue from this point. Okay. Have a good day.